on YouTube today. There we go. Great. Okay. Welcome back to day 2, part 3, where we're going to talk about annotation. And so we're just going to dive right into this. I have a little bit of a spiel. Like I said earlier in one of the other sessions, there's not as much hands-on right here. We're going to do a couple things with a local blast search and a local HMM search to kind of get a feel for what that is. I'm going to start right here because I know JP is going to ask me this when we get a little further into it. And it's going to be the question of how do you know you've perfectly annotated your data? And I'm going to tell you right now, you don't know. And that's going to be, I'm spoiling the surprise at the end of this. And it's not on the first slide, but maybe it should be, which is that annotation is a hard process, right? There's a lot that goes into this and a lot of assumptions that happen. And there's a lot of room for error. And so like a lot of things that we're dealing with when we're reconstructing biological data in a computer system, in a computer environment, we're getting our best guessed approximation of what biology is doing. There's a reason why for a really long time, metagenomics and these omic data sets, there have been elements of our larger microbial community who have been like, well, yeah, but you don't know that's really what's happening, right? The real gold standard is still getting an organism, doing an experiment, getting a protein, crystallizing it, getting some information about function, right? So all we can do is work with the databases that we have and the information that we have and try and get at a best guess approximation of where we're going from here. So I'm going to start with that. I'm going to spoil the ending. This is hard and there's no right answer. So let's have a bunch of fun trying to figure out some answers along the way. So that's going to start with the premise of this whole section is going to be on. It's like, maybe we won't learn anything. We'll learn something, but maybe not anything. Okay, so our goal with annotation really, right? And so we touched on this in the assembly part of this, is to find the features of interest in our genome, right? And so we talked about feature identification in that assembly point, right? Where we were looking for non-coding elements like the rRNAs and tRNAs and our protein coding sequences that we worked with Prodigal and any of the extra stuff that we might be looking for on top of that, like CRISPRs and insertion elements. There's a lot of ways that the name that we look at actually gives us some information about how confident we are about a protein. So if you're on NCBI and you have these, so there's some examples down here about subcellular location and domain structure. The names that are in NCBI, when you get them, have a structure to them, and they are indicating how confident we are and where these proteins are being processed and put into the cell. So they are concise names, but they're not necessarily really descriptive and super informative. And so there's lots of places that we can go looking for functional annotation, right? So just looking for the finding the elements before we do anything about assigning, for feature production, before we get to the functional annotation part of it, there's a lot of places that we can go. And then once we have the elements that we're looking for, so we have run Prodigal and we have proteins to look at, we need to find some way of switching from feature prediction to function prediction. And so really what we've been doing for a really long time is using homology as a predictor for function. And so this has gotten easier as the databases increase in size. So this was assessed back in 2015. So we can all assume that the number is much larger now. But if we looked at the total number of bases in GenBank, we're talking about over 1 trillion bases of possible matches that we can be looking for, both in protein space and genome space, right? So there's a lot of data out there. I'm sure this number, I don't know what it is right now. I'm sure it's at least twice that, if not more than that at this point. All right, so there's a lot of ways that we can do these searches. The one that I think a lot of people are most commonly associated with, this is maybe something that you've done even as far back as undergrad, is working with Blast and looking at local alignments or pairwise alignments. So there's a little example here of what that might look like. So on a Blast search on NCBI, you get this setup where you have these stretches of, in the case of protein, and you're looking for matches and similarity between your target your query sequence and then some target in the database. And really what you're looking for with these similarities is a best hit, right? Something that matches it as closely as possible in the database that exists already. And so, like I said, you do this using something like Blast, and it's implemented in a lot of different ways. And we'll do some local blasting to answer these kind of questions ourselves. There are lots of databases to do this with. So NCBI has a number of databases, like the non-redundant database that includes every protein, every sequence that has been submitted in a non-redundant format. There's RefSeq, which is a curated database of microbial genomes and other genomes that exist. Databases like UniRef, databases like KEG, which is the Kyoto Encyclopedia of Genes and Genomes. And so you can move up. There are these different databases that have these different functions. So if NCBI has everything, and you can blast or take your sequence and do a comparison and search it against everything that maybe has ever been discovered and submitted by the scientific public, KEG, on the opposite end of it, has a much more highly curated database, which takes all of the proteins that they know of and put them onto chemical maps and chemical reactions, so that there's some information beyond, oh, it's a match to a hypothetical protein from some organism that we've never actually seen any function for, right? And so this difference in database is really kind of how we approach this. What is your target goal for working with this type of data? Is your target to find out if your protein that you're looking at has ever been seen before? Nowadays, especially if you're looking at hypothetical proteins, that might not be the most exciting question that you can ask. It can be. There's definitely ways you can make that an exciting question. And then if you want to know something about, okay, what elements of a genome am I looking at that I can assign to some type of particular pathway or function or process, you might need something that's a little bit more highly curated like KEG. And so to give you an example of what KEG looks like, here's a KEG map for methane metabolism. And so here would be every one of these boxes here with the numbers in it represent an EC number, right? So this is a number that tells you a little bit about the functionality of the protein you're looking at. And you can use KEG to find, elements of these pathways in your genomes. And when you do, they'll start to be, you can use several different systems to overlay this over this type of map and see which elements of these pathways exist in your genome. We'll come back to that in a little bit. So what we want to be doing in a lot of these situations is detecting areas of similarity. And so one way is that we can do this much more specifically and moving away from a general alignment using blasts. And we'll get into a little bit more details in this, is using something like a hidden Markov model or an HMM. And what we do with an HMM, it requires some information before we get started. But what we can do is, instead of looking for this very large similarity search, can we find a sequence that looks exactly like the element that we're looking at? What we can do is we can build models based off of alignments of genes that we think have the same function, or we know definitely have the same function. We can make these alignments that look like this, and then look for regions that are highly conserved and build models off of those highly conserved regions. And this allows us a little bit of flexibility, because then we can use the models to do our searching as opposed to looking for broad similarity matches. And those models allow us to find regions that are important. So in this case, they might be like regions that are the active site or regions that are important to structure. And you can look for those specifically as opposed to just some broad similarity. And that would give you some higher confidence that what you're looking at, one, has some of the similar functionality that you're hoping that it might in terms of the prediction. And two, it allows us to actually look for things that are slightly more divergent than we're just looking for something that looks exactly identical over some stretch of our reference or query, right? So you can use models to like find very specific functionality in things that are much more divergent than just basic blast searches. And so in this idea of making an effective profile, what you have to do is you have to start with seed proteins, right? So this takes a little bit of time. It's really nice when somebody's done this for you. So there are a couple of databases that we'll talk about that have done this for you, but you can do this yourself. You can get a collection of proteins, you can align them, you can trim them. And once you have a good alignment of, with a high confidence that what you're looking at are actually proteins with the same function, you can apply a tool called Hammer to these alignments and make HMM models based off of those alignments. And it will find the regions that have the most conservation, and you can use those as your own model. So you can make your own versions of this and then test them and look at some various cutoff values, right? So when the model is created, it uses some statistical probabilities to generate what they believe based off of the model would be an effective cutoff score when you do a search, but you can always play with this, right? So if you lower the cutoff score, you can actually make it so that you're looking for things that are slightly more divergent than what you started with. Okay, so there are lots of places where you can look for HMMs to work with. And so one of those places is the COG database, where there's this collection of genes that have been collected that have the similar functionality for some core metabolisms. It has to be in at least 3 genomes. They've been manually curated. For a while, these were created and then weren't maintained, and then are now back to being maintained. So they actually offer an avenue for doing this. Things like TigerFam, which is has been generated but no longer is maintained. So you can go back to it. That has some very high confidence, manually curated, a subset of functions that have models for it. You can then go to PFAM, which is a database of HMMs that highlights specific modules, right? So not the whole length of a protein all the time, but like key domains. And when you have multiple key domains from PFAM aligned to a protein, you might be able to say something, especially if there wasn't a function to begin with. One thing that's nice is that TigerFam is incorporated into PFAM. So some subset of the PFAM HMMs are actually these full length proteins that TigerFam has, but then most of the elements in PFAM are domain specific. There's also this database called Foam that's partially complete, where they've taken all of the terms from KEGG and made HMMs where they can based off of the KEGG pathways. That doesn't have all of the elements in it. There's some information here that you can always follow up on that we'll talk about a little bit. So the difference between an equivalog, right? So this is something that is a full length an alignment in the HMM, and all members share that same function. So you have a lot of confidence in what you're doing with the model. And then because the model has high confidence, you know that the match is high confidence. This super family, right, which is you have proteins that potentially all have the same function, but you don't know for sure, but they do share a lot of the same structural format. They have a lot of the same helices, they have a lot of the same areas around the active site that look important. We don't necessarily know that the function is conserved there, but we do know that this is a family of proteins that might have some linked function between them. And then we get into these much smaller regions that I've alluded to in PFAMs before, which are domains, which highlight really specific areas of a protein. And the example here is ATP binding, right? So these are modules that we can see in multiple proteins and know that there's a function in that protein that does something, like in this case, binding ATP. But in some cases, that might be a hypothetical protein. So we know that it binds ATP, but then we don't know what the rest of it does, right? So that it gives us some information. It allows us to go down a rabbit hole a little bit about how hypothetical proteins, if you're working with one of the many organisms that has less than 30% annotatable genes, this is an avenue for you to take where you can start to look at and maybe conceive of ways of how these proteins might be functioning in the cell. And so this is always a good reminder at this point in time to tell you that you can get matches for proteins that don't have the same function. And so this is just a quick reminder from basic biology, the difference between a homolog, an ortholog, and a paralog, where homologs have some similarity between very broadly based proteins, but only orthologs have similar functionality with each other, right? And so in this evolutionary example of hemoglobin and looking at the alpha and beta chain, so this is a very, intro bio example of this situation, but very good to rely on. You have this setup where all the orthologs on one side of the tree for the beta chain have the same function, but look very similar to the alpha chain of orthologs for the hemoglobin here, which also have their own function. So collectively, within the functionality process, right? So we know that these all have the same function. If we look at them compared to each other, we call these paralogs. So even though they look like each other and they would match each other in some type of similarity match, they actually have different functions. And because they're all descended from the same early gene, we just call them homologs. So, there's this hierarchy of terms that we can use that give us some output of the idea of how confident we are and what we're looking at in terms of functionality, and put an evolutionary spin on it as well. And so, just to reiterate what that actually means is that it means that you can get matches between proteins that have different functions. And that's always something to look out for. And so when you do something like a BLAST search, really what you're always looking at, or HMM searches for that matter, what you're really only confident of is that you have homology between your proteins, and you don't know you have orthology between your proteins until you make a tree of it. And so because homology doesn't equal function, there are lots of problems that we know about where we can have issues. So the first one's very easy. We have a query, there's a template that we're matching or a reference that we're matching it, and they're paralogs of each other. So they look similar, but they don't have the same function. Or we have this example here, this moonlighting problem, as it's referred to, I guess, in the comp bio world, where we have a query and a template, but that template has more than one function. Right? So while it's really great that our query matches it, we don't actually know which of the functions that our query might have because the templates that it's matching to have different functions. We have this idea that your query sequence might match a portion of a protein. And so this could be because they have similar domains, and those domains might have similar functionality. But if the elements of this part of the protein do something different than this part, then you can end up in this situation where you're predicting functionality that you don't have in your query. And then last but not least, and this is one of the really bad downsides of a public system tragedy of the commons situation, is that the template that you match to might be misannotated. We relied a lot of time on people looking at these sequences and going through. And so if there's an error that was made someplace back in time, and no one's ever corrected it, that error will just continue to propagate in the database going forward. And so we know a lot about this, right? So here's some examples are, there could be errors in the sequences themselves. So either actual sequencing errors, right? So we thought there was an A and there's actually a T, and that changes the amino acid that's there. So that's not actually what the protein looks like in the real world. People like to keep their cloning vectors in their sequence. So sometimes when you do your protein predictions, you'll convert your cloning vector into a protein. The actual gene calling errors, right? So we talked about this earlier today, where you could actually predict proteins within an rRNA. You can make it, there's a gene call there, but it's a gene call error, right? And if that gets into the database, which it used to, you can have that problem going on there. And then you have this idea that because when we did these annotations originally, at some point, they were incorrect, but they're still in a system, you have this genome rot, where there are these inaccuracies or mistakes that continue to propagate forward in time. And so we talked about a bunch of databases back here. This Oxford Journals link right here actually has a link to a bunch of genomic databases that you can, when you have time, you can click on and you can look at and see which ones I haven't covered and get more details on the ones I have. So then you get into this situation of what happens when there's no similarity. What happens when you have something that has no homology to something in the database, or everything that you have homology to in the database is a hypothetical protein? So this is when we can start getting into, there are multiple tools out there that allow us to try and get into this information. One of them is PFAM, right? So we can run it against PFAM. We can look for domains that might not explain the full functionality of our protein, but might give us some information about what it can and can't do just based off of that information. We can look at our hypothetical protein in relation to all of the other genes in its proximity. And so a lot of the times in microorganisms, bacteria and archaea, what we have are operonic structures, right, where the whole area of the genome is expressed and converted into protein at one time. And it's generally because all of those proteins serve a function in some pathway together. Right. So if you're looking at something that looks like an operon and you have some form of annotation in there that's all nitrogen based, you might be able to say something like, oh, this hypothetical protein might have something to do with this nitrogen pathway that I'm looking at. That becomes very difficult to make the argument for reviewers, but it might give you some idea for, if this is a really important pathway that you're looking into and you have this organism in culture or you have this organism in enrichment, how can you figure out a way to either overly express this protein or find this transcript being expressed so that you can get at that information more accurately. Oh, there's a question from the webinar people. So this is a question from Darcy on the webinar. Will you be addressing how one might avoid being fooled by information that hasn't been annotated correctly? So this is one of those things, it's more of a warning than it is anything else. What you can do a lot of the times with things that have been misannotated is you get at it a little bit more confidently if what you're looking at has overlap between multiple databases, right? So there's lots of overlaps between databases, not 100%. Otherwise, why would you make a new database if it's exactly a copy of something else? But you can use these comparisons. So like, you can compare your results that you get from NCBI, you can compare it to KEGG, and you can compare it to other databases like Uniprot or other databases like that. I'm not going to list them all because we just could be here for all day. And if what you're getting are matches to functionality in all 3 of those databases or all 5 of the databases that you're looking at, then you can be a little bit more confident that what is in there has not been misannotated. If you go into NCBI and you get one result and it tells you that it's a reductase of some kind, and then you get into all these other databases and it's telling you that it's a transporter, that seems like there was an error in one of those cases. And you have to then make a decision based off of the efforts that you've put into this. Do you trust the multiple databases that have the same annotation? If everything is different, what can you do in terms of looking at the structures of the matches that they're telling you are matches? What can you do on your own, looking at alignments, looking at trees to see if that makes sense? If you don't like making trees, looking for like really high specific confidence in your functionality is going to get really tedious to you, because it's going to be a lot of tree making, a lot of alignments, and a lot of confirming results that you really hope would just be done automatically. But the more confident that you want to be, or the more of a stand you want to take on a particular function, the more effort that you're going to need to put into proving that through various other methods than just, I put it in a pipeline and this is the result that came out. Not that that doesn't work. You can get really broad answers from a pipeline that are really helpful and really good. And the more broad you are, the less accurate. You don't have to make a tree for every gene in your database or in your sets of genomes that you're working with. That would be crazy. No one's expecting you to do that. But there is some middle ground that you'll have to tread to do that. Other things that you can do if you don't have any homology, you can ask the question of where is this protein being transported to within the cell. And so there are tools like pSort-B, which give you an approximation of cell localization. So if you have a protein, it's being exported to the exterior of the cell or as an extracellular protein, that might give you some information that you previously didn't have about what type of function is happening there. There are topological features that you can look for. So methylation sites, sites that might be binding to different types of phospholipids. And then there are predictions for various types of very highly specific sets of databases. So one of the examples that I have here is the CASI database. And so CASI is short for carbohydrate active enzymes. And so this is a database specifically designed for using HMMs, not HMM, but there's an HMM element to it, but more accurately finding proteins that have a specific carbohydrate-based functionality that then you can look at that might not be its general function. It might give you, or a specific function might give you a general function, but you can use that to find things like carbohydrate binding motifs or hydrolase activity that might give you an indication of the type of carbohydrate that it's interacting with. And so there's a couple other databases like that. Another example of that would be the MIROPS database, which does that for peptidases. And so these are highly specific databases that are somewhat manually curated at this point. Some of them are automated as well. They're elements of both that give you specific functionality outside of the realm of homology that might not match in a database. Yeah.
What did you say was the peptidase database?
MIROPS, MEROPS. Okay. So I've given you all this information about databases. I haven't given you any examples about how you could do this automatedly. You know, I'm sorry about that. That's maybe a topic that we'll have to get into a little bit further down the line. But let's say you have an organism that you're working with. And really what you want to do is find specific elements that you know should be there, and you want to work with them specifically. So one of the ways you can do that is, you know, setting up and performing your own BLAST search, right? And so in this particular example that we're going to use, this is an organism that we were able to generate from the Tara Oceans metagenome data set. So this is an environmental organism. I can tell you that we believe it to be a cyanobacteria. And so we're going to use BLAST to search a local database of highly confident genes that we are highly confident are part of the carbon fixation pathway, the Calvin cycle, actually all of the carbon fixation pathways. So it's a highly curated manual database where I went in and I found proteins that had high confidence of being the function that we were looking for, put them all together, and then we can use BLAST to search the predicted proteins for this genome and search against that database and ask, well, what type of carbon fixation is this cyanobacteria doing? Well, also, and so this is like a little bit of a precursor for what we're going to do with the phylogenetic side of it, is we can use HMMs. So we'll do BLAST first, and then we'll move down to HMMs. We can use HMMs to search for ribosomal proteins, specifically the ones that were used to make the hug tree in 2016. So these will be 16 ribosomal marker genes that most organisms have, and we can use HMMs to pull those out and do that. So those are the two examples that we're going to work through. Yeah, so we're going to do these locally. Why? Because custom databases, you know, especially if it's your custom database, has gone through a higher degree of curation. And small databases, once you have a small database, run quickly, right? So you don't have to take your data and compare it against some big database, compare it against all of NCBI, compare it against all of KEGG or something like that. You can be like, okay, well, I have a new genome. Today's a new genome day. Let's see what we got. I can do a quick blast against all the carbon fixation pathway genes and see, do any of them match? It's that kind of idea about why we might want to do this. So we're going to use BLAST. And so this is where we're going to get a little bit hands-on. And so this is the Wikipedia definition of what BLAST is. It stands for Basic Local Alignment Search Tool. And it allows us to find these elements that have similarity across organisms or across sequences to find matches between sequences. And so in this example, we're highlighting sequence 1 and sequence 2. All of the matches have these lines between them with little pluses. And then we have two different areas that we're looking at. We're having, we look at a mismatch, right? So this is just a single area where there's a difference between the sequences that we're comparing, and then a gap, which is a stretch of mismatches, either in the sense that in this particular instance, it means that there are sequences in one, there's a stretch of sequence in one sequence that isn't present in the other one. All right. All right, so we have this genome. It comes from the Mediterranean. We're pretty sure it's a cyanobacteria. It has 35 contigs, has 1,500 putative proteins based off of protocol, and we think it's about 2/3 of the way complete with a little bit of redundancy. So we're going to compare this example genome to a database that consists of all of the marker genes for the Calvin cycle, the Wood-Lungdahl cycle, the reductive TCA cycle, the 3-hydroxypropionate cycle, and the 3-hydroxypropionate 4-hydroxybutyrate cycle. Does anybody want to take any guesses before we start about what pathway the cyanobacteria poly is fixing carbon with? This is not a trick question. CBD. Yeah, probably. All right, so that's what we're going to be doing. We're going to do that now. So if we start here in our base directory, so right now I'm at home C-DEBI, we can move into EcoGeo, and then we can move into the annotation directory. So if we hit ls in here, we have a couple things that we're working with. So this is our example genome in FASTA format. It's our example genomes proteins. And then we have a database called carbon fixation markers.ffa. So this is just a protein database. And then we have a database that is the hug ribosomal markers in HMM format. So we'll get to those shortly. So to effectively do this, what we need to do is first, we need to index our database that we're going to be working with. So we're going to use a command called make blastdb. So this is going to make a blast database to work with. So we can do that together. It's going to be make blastdb. We're going to do -n. We're going to tab out carbon fixation. And then we have to tell it what type of molecule we're working with, because computers are dumb. We have to hold their hand for everything. And so that's going to be -db type. And then we're going to type prot because it's a protein database. That's a really good way to get murdered, right? Is to say that about computers on YouTube so that when they become sentient, you know, this could be a horrible, horrible mistake that I just made there. Future computer overlords, I didn't mean that. So if we hit enter here, it should run pretty quickly. And so what it's done is it's indexed our proteins that we were looking at. So there were 325 in our database. If we hit ls, you'll see that we've created a whole bunch of extra files. And so this works, we need to do this no matter what size of the database we're working with, but it's particularly as important once the database gets very large. The index allows it to make really quick searches as opposed to having to search through everything individually. So it's like a lot of shortcuts. So Blast, if you're not familiar with it on a local setting, has lots of different types of outputs. So the common output, the one that you would see if you were going to NCBI, would be this 0, the default one. And so this is going to print out some very elaborate examples of similarity, right? You'll get a match. Your match will have some alignment. It's going to list all of the proteins that match matches to. It's going to give you a whole bunch of scores. It's very useful, but it's more useful on a web server or a screen that you can scroll through and click on things as opposed to trying to digest either as a person or, and as we're going to hope to do, using the computer. So we're actually going to use one of the other built-in options here for generating an output. It's called the tabular output. So this is going to generate, it's going to take all the information that's normally displayed in a slightly graphical form with the little lines between things that match and pluses and minuses. And what it's going to do is it's going to convert all that to just the raw numbers. And we're going to get numbers out that are like where does the query match to the subject? What's the score on that? How much percent identity do we get? How much percent similarity do we get? Because these are proteins that we're going to be looking at. So it'll be all summarized in a tabular output table. So we can run this together. And so this is going to be the command that we run. We're going to run Blastp because these are two protein databases. We're taking our protein queries against a protein database. There are lots of other options for Blast. You can do nucleotide versus nucleotide, and then other versions of translated nucleotides versus proteins in multiple different avenues, depending on which is which in your database versus your query. And then we're just going to spell it out. So it's going to be blastp-query. We're going to use our Tara Med genome that we're going to tab out, the ORFs. Our database is going to be the carbon fixation one that we have sitting there. We're going to print it out into a temp file because why not? We're going to set an E value. So this is one of those elements where we get to start to be a little bit more discerning about what we're doing. So an E value is this prediction that Blast will input about how statistically likely is this an actual match versus an erroneous match. And so this is dependent. This value actually changes in terms of what value you want to use here based off of the size of the database. So something like NCBI RefSeq, which has, or NCBI NR, which has millions and billions and trillions of proteins in there, you can actually use a much smaller value here, or in this case, a much larger value. 10 to the -5th would be fine in that type of situation to find a fairly similar match. And the reason that is because the chance of a match in a database that's that large just by random chance is actually, statistically fairly low. And so if you have a really good stretch of gene homology, and it gets a really high score, you can have a lower value. In this situation where we only have 325 proteins that we're searching against, that match or a stretch of a match might be enough to trigger a response because it's making these comparisons between all of the results. So what we want to do here is actually use a much smaller cutoff value, e to the 1e to the negative 20th, right? So this is saying we don't really want all these little tiny matches between these proteins, only find us a statistically good long match for this limited amount of data that we have. And then we need to limit some outputs because otherwise it'll find all of the instances of all types of overlaps, and we don't want to do that. So we're going to do dash num underscore descriptions and set that as 5. So we're only going to get 5 outputs per input. And we're going to do it. So it requires setting both num descriptions and num alignments to 5. And then if we hit enter. All right, so that ran through. So you notice here that we haven't set the tabular output. So this is the reason why this is temp is because we're just going to be looking at what the standard output looks like. And so the people who are working on this for the EcoGeo side of it have highlighted an area that we can use. So if we use less and then add a number using this plus sign to it. So in this case, we're adding a line number, 1,100 or 11,096 to this temp. It's going to take us to that line in particular. And if we're lucky, it'll be this region that's shown in the picture here. I can make this a little bigger. So this is that visual output I was telling you about. So this is just the standard. So here is succinyl-CoA reductase was the input as part of the database. And here is the match. And so here we're looking at proteins. And so what we're seeing here is a graphical, graphical used lightly here, representation of how good the alignment is. And so everywhere you see a letter between the two letters here, so this example right here, GKP, so this is a direct match in the amino acid sequence. If you see a plus, it means that these are amino acids that come from the same family of amino acids. So they're similar to each other, right? So they're either the same size or the same polarity. So there might not be that much of a difference in terms of how it impacts the structure of the protein. And we can scroll down and look through this and we get all these results. And at the end, there's some information from the BLAST output. And then we get into the next query. And that for the query that we're looking at here doesn't have a match. So we can go back up for a second. So this was had a match. There's some information here that we can go through. So this is a bit score. So this is a representation of our E value plus how good the matches are in terms of sequence similarity and identity. And so a bit score of 98 is not great. You can see here that for the identical matches, so how many times are the letters 100% matched up? We only have 107 matches out of a total of 408 total amino acids in the protein. So our identity value is fairly low, 26%. But if we ask all the places that have identity and are positive matches, so they're similar, so this would be known as your percent similarity, we can look and we see it's a little higher, it's a little closer to 50%, where you have 171 out of 408 are either identical or similar to each other in terms of their amino acid positioning or amino acid type. And so what does this mean, right? For this particular example, is this a good match or a bad match? And JP, I have some bad news here for you. This is coming out to you. I have some bad news. Is that protein homology, we've talked about this through this session already, right? It's it doesn't describe function very well. It allows us to have these matches. In general, we can have these assumptions that proteins that have an identity are identical to each other or similar to each other, depending on the protein, at about 35% similarity might have the same function. At the same time, proteins that are 99 or 96% identical to each other might have different functions. So we run into this really, so what does this mean? So this means this is not a great match, is what this means. And that potentially it's just the fact that, and this is why there's a couple versions of this, it might be that this succinyl-CoA reductase is just divergent enough from the cyanobacteria that the match is very low. And then if we looked at a couple other ones, we'd be more confident that this protein is what we think it is. But at the same time, if this was the only evidence that we had that this protein was a succinyl-CoA reductase, we might be suspicious about what the actual function is of that protein. We can say that it's related to it and that there's homology, but we can't say for certain what it does. Yep. The what? I'm sorry. Oh, so that's the E value. So that the expected value. So the matching E. So we set it at -20. So this match had an E value of -24. So again, it's not a bad match. It's not terrible. If you're grasping at straws, you'd be really excited about it. So we're going to try this again. We're going to run the same search, but this time we're going to simplify it using the tabular output. And so this defines, so in the tabular output, there are lots of ways you can change the options around, right? And they all have these different names. There's a website for this, which is not linked here. But if you Google Blast options, will come up. And so there are these abbreviations that you can add or remove that will give you extra information that may or may not be part of the default. And so we're going to work with the tab output. And so you can see here, maybe not, I can make it a little bigger. So what we're going to do is we're going to do an output format that's tabular, and then we're going to only ask for certain elements of that tab output. And so in this case, we're looking for a query seek ID. Where does the match start for the query? Where does it end? This subject ID, how long is the subject? Where does that match start? Where does it end? What is our bit score? What is our percent identity? And what is our E value output? And so I, like you, should not try and type out all of that information. We'll copy this one like a bunch of cheaters. And that'll run. Oh, okay. Oh, that was my fault here. There's an extra return in the PowerPoint. Nope. That's not what I wanted to do. So if you're running this, you need to look for both of them. I'll highlight this in a second. Oh, and there's two here. Great. Okay, so the problem with this is that this has added an extra return here. So if you go into the PowerPoint, if you're copying from the PowerPoint and you go back, just hit delete at the beginning of this dash for max, it's going to remove that space. You can see that now instead of a return, there's a space there. So if you make that small little change and now copy it, it'll run correctly on the computer. Is anybody having problems with that? We can solve it for you. I'm going to come around. I'll be back, webinar people. Too many positional arguments. You want to go back to the PowerPoint? So you hit delete here, right? So there's no longer a line. Yeah. What if we try and type this one all the way? Okay, let's try that.
Can you switch back so we can see what?
Yeah.
Of course.
So let's try that. That's going to take a little bit of time. We can do that. It worked for you guys on this side.
Yeah. I didn't type it up. You shouldn't need to do it.
Great. Okay.
I started typing it.
I'm working on one here for everybody. It's just Sarah that we're having a problem with. Nope. Great. Oh. We solved it. That minus 20. Great. So we're just waiting for everybody to catch up here in person. Dan, I'll say this time. This is why I said don't type it. I know. That's why we're trying to skip all this. Right. Okay. So that's perfect. All right, so we can take a look at this information as it comes out. All of you who have cheated and started to get your snack early. We're going to move on without you, so you'll just have to listen. So if we do less here and look at the name of the file that we were working with, right, which we named in this particular output as opposed to a temp file, we're going to call it, we called it blast_output.tab. This is just to indicate, one, that we ran a blast search. This is the output of it, and that it's in the tab format. And so we can look at it, and there we get there's a lot of numbers in columns. And so I've already heard a couple people here at the table closest to me commenting on, well, that's not really that useful. If I'm looking at this, how am I supposed to know what these numbers are? They're all over the place. I mean, I guess I know that's an e-value and that might be % identity, but what are we supposed, like, how am I supposed to work with this? The idea is that this tab format is not for you. So the visual format that we looked at, that was for everybody, right? We looked at it, we could see what the alignments look like. This tab format is for the computer, right? So this allows us to do like, instead of what we had originally, where you had this like really intricate output with equal signs and names and lines varied, and sometimes you had a hit and sometimes you didn't have a hit, and there's all this text. Now all we have is this brief summary of what the results are, and we can do things like, which on the next slide we'll do, we can order these based off of match scores. We can find out the information that way and then figure out meaning off of that, right? So if we go to the next slide, not to call you guys out, but just to iterate to the crowd why, despite looking awful, I'm telling you that it's the best thing that we could possibly have is this tab output.
Can you not add headers like yourself?
You can, yes. So if you wanted to add headers, you can go in and you could put tabbed in headers and have all that information. And there's, if you have a small enough set of information, you can take that information then directly and put it into Excel or something like that, which is a dreaded word for bioinformaticians, but we're all biologists, so don't tell any of them that I said that. Okay, so if we wanted to ask for every element that was greater than 50%, right? So there's some information here in this figure. I'll pull this up again real quick. I'm gonna do ahead. So if we were to count where the columns are, so this is 1, 2, 3, 4, 5, 6, 7, 8, 9. In this particular instance, 9 is our percent identity value here. And so this is one of those things where I used to do this really the hard way for a really long period of time. And then the person who we asked to do this part of the workshop for EcoGeo was like, why don't you just use the command line to solve this? If you want people to find where everything's greater than 50%, just have them do that. I was like, great, show me how to do that. And so we're going to use a command that we didn't really talk about yesterday at all in the Unix portion, but it's called awk. It works slightly similar to the way that sed works, but not quite the same way. It allows us to essentially give Unix something that we're looking for inside of a file, and then it can find the output for us. So if we were to ask that question, I'm going to make this smaller for a second, of, can we find all of the elements that had a match of greater than 50%? Because, and if you can see right here in this little bit of area that's visible, it looks like a lot of the matches that we find are in that 30% range. And that 30% range, like I said, doesn't have a lot of biological meaning all the time. It's something that can happen just kind of by the fact that a lot of proteins look like each other. And so even though there is a match, it might not be a strong match. Once we get into that 50% range identity or something higher than that, something which we'll get to in a second, into the 90s, then we can start feeling a little bit like, so this is a correct identification of the proteins that we're working with. Okay, so we can type this out. We can type out awk. You have to be careful here if you're going to copy and paste, because those have been created into special quotes as opposed to just generic quotes. We're going to do a curly brace if, open parentheses, $9. So we're telling it to look in the 9th position of the columns. If that number is greater than 50, we want it to print. So that's just simple command statement there. We can close the curly brace, close the quote, and then we're going to say look in the blast file for that. So it's just awk, a little bit of information. And then if we hit enter here, so we can find anything that's greater than 50, and there is not a lot that's greater than 50. There's just a handful. And not only is there not a lot greater than 50, there's not a lot between that 50 and 75 marker range. And so,
the zeros with the E-score?
So when you have an almost perfect match with your E value, it drops to 0, right? So it keeps getting, so as it gets smaller, so, negative 20 is a small number, negative 100 is a small number. It gets to the point where it doesn't add anymore, and he just says, it's a perfect match, or a really high.
Zero, so
yeah, right.
Like something wrong.
So that's a good thing, the sign of a 0 for the E value. So if we do that, we can now do something like, so this is information is all kind of like mixed around. We can bring back up our last command, hitting the up button, and we can pipe it, image. We can pipe it to sort, and then throw in a couple flags here that we're dealing with numbers, and we'd like to order it in a specific way. And we're going to work on specifically field 9. That's where all this information is. And if we hit enter, now the same data is sorted for us based off of percent identity, where we have the top match being a 91% identical protein for this particular protein. And so now we need to know, right now all we have is a subject ID about what this protein might be, this top match to the proteins that we've been looking at in this file. Yeah, it's column 9, and then I have to double check what the comma 9 is. It's column 9, and then sorting by that particular element too. Yeah, I have to double check that for you. So what we're going to do here is we're going to take this particular ID that we're working with. We're going to copy it, highlight and copy, and then we can use grep to search in the carbon fixation marker gene file to figure out what this ID belongs to, because there's actually some more information in the header line than just the name. If we hit enter, we find out that it's rubisco, the large subunit, which is good. That's great, right? We guessed that cyanobacteria would have Calvin cycle, and it had the Calvin cycle. We win. Go biology. And so this is, something that you can do obviously on your own with whatever function of interest you want. If you're interested in arsenate, if you're interested in iron, if you're interested in nitrogen, you can have these kind of little databases that you've made that you know are 100% accurate, or 80% accurate, or 90% accurate, depending on how much time you want to put into it, and then use them to search anything new that you come along really quickly, right? You don't have to wait for that big data set to come back. You submit your genomes to IMG and it takes 3 weeks to process, or you submit your data to MGRAS and it takes 3 weeks to process. This is a good way of trying to get an answer, and a high confidence answer. So even when you get a result back from IMG or MGRAS, you can then use this, these kind of smaller curated databases to be sure that the results you're getting back are real. And then we're slowly rounding out the day. So now we're going to talk about how to use an HMM database. And so just as a quick reminder, what are HMMs, right? So this is, we're going to use a tool called Hammer, which is looking for homology based off of the hidden Markov models that have been generated for a particular protein or not. And so this is another way of looking at the way a Markov model might be interpreted. So these values under here are some scoring system that they have. And here they're ranking how often a certain amino acid is in a particular position. So the larger it is, the more times it occurs in a sample or in a sequence at that position than other amino acids, right? So in this particular case, these Ys are conserved, right? They're there in every sequence. Here, L happens much more frequently than I, while at this position, there doesn't seem to be a lot of conservation of that particular amino acid because it's shared by a bunch of different amino acids. So if you're going to do this yourself, what you need to do is you want to make an alignment file. Like we said, you want to be very highly specific in terms of the level of curation you're going to do that. You want to make it as nice as possible of an alignment that you can get. And then what you can do is you can supply that alignment file to Hammer, this program, and use the command HMM build, which we're not going to do here, and generate a new HMM profile. And so you can do this for proteins that so I think one of the really good examples of this one that I can think of off the top of my head are some of the proteins that come out of the Zeta proteobacteria for iron oxidation, where we have like a fairly poor idea consistently of what those proteins may be that have that function in them in the iron oxidation metabolism. Jeff might argue with me one way or the other. Totally agree. It's great. And so, as we've gotten more and more Zeta proteobacteria into culture, Roman Barco here at USC, who the RUs work with, has had some luck doing some protein extractions, and he was able to try and tease apart which proteins were interacting with iron in the extracellular part of the membrane, and was able to target a couple extra proteins that we didn't really think were part of the electron transport chain of this particular Zeta proteobacteria. And so, that's great. And you can do these homology searches where you can take a new candidate and blast it against stuff and look for things, or we could build an HMM model for that. And so that HMM model doesn't exist. It's not in the database, but we could get a couple of these proteins that we think have similar homology, make some judgment calls, and then build a model, and then use that model to search against a larger database in a little bit more of a more high throughput, higher confidence way of figuring that out. So that would just be an example that I've personally worked with, but you can do that the same, right? So this might not be ideal for the NIF-H gene that probably has 100 models from every group that's ever wanted to do something like this. But it would be useful if there was a particular sub-process that you were interested in very specifically. Building a model off of things definitely have that functionality can actually be useful, because then you can have something that is easier to search and with a little bit more confidence than a simple similarity search. So I mentioned before a couple of the HMM databases that are out there, including PFAM, TigerFAM, and FOAM. And so what we're going to do is we're going to use a database of the hug ribosomal marker proteins, and we're going to identify all of those in our putative cyanobacteria. So we're going to do that using Hammer, like I said. Let me make this a little bit. All right, and so we can do that by calling Hammer Search, which is a tool within Hammer. Oops, misspelling table out. So this is a version table out. We're going to call it HMM output .tab. We're going to tell it to use the trusted cutoffs, right? So these are cutoffs that when the model is created, Hammer has decided that these are the model. If you have a score above this value is something that we'll keep. If you have it below this value, we don't want it. We're going to do some information here where we tell it in terms of the output. We don't want to see any extra text except for the raw output. That's what we're working with. We're going to tell it which database we're working with, which is the Hug ribosomal in our folder. And then we're going to compare it to the Mediterranean orfs.ffa file. And then if we hit enter here, it should run. And it runs really quickly and does this stuff because there's not that much to search. And then we can look in the output here. And this has a header for those of us who wanted a header in the last one. And we can look at the results here, which are going to be tough with the view that we have here. So this is one line spread across multiple lines because of the view. But what we have is a protein ID. And then because the names are named the way that they are in the search file, we know that this is the ribosomal protein RPL5. This is the PFAM that was associated with that. We have a e-value score. So the e-value here is a similar metric to the one from Blast, but doesn't have the same exact meaning. So you can look at them, and if you know what an e-value in Blast means, you know what an e-value in your HMM output means, but they're not 100% directly comparable. So, you know, an HMM value of -20 and an e-value or a Blast value of -20, while you can, you know what that means, you don't say, oh, one's better than the other. You can just, they're calculated in a similar way. We get some extra scores. It breaks it down based off of the full sequence and the best domain match. And there's some more information that you can look up here. But essentially what we've been able to do here, I'm going to try and see if I can get this all in one spot. If I do it like this, and you look down this list right here, is we've identified marker proteins. And if you count this, you'll find that there's more than 16, which is interesting. But there's actually a couple duplicates here. And so this is RPL22 is duplicated, RPS19 is duplicated, but that's it. And that's troubling. But if you remember back when I was giving you some of the statistics about this particular genome, we believe it's about 2.4% contaminated. And so these marker proteins here that have duplicates are probably the result of that little bit of contamination. So it would be something to go back in, maybe with the NVO. I don't know why I would have shown you all that information before this, where you could go back in and make some adjustments to that genome and make it a little cleaner so that this works. At this point, and this is what we're going to do in the next session with the phylogenetic side of it, we could take all of these markers and start using them to build them into a larger phylogenetic tree on a big tree of life, right? And get an accurate idea of what type of cyanobacteria we're looking at. So then as we round out this section, I'm going to remind everybody that annotation is hard, that biochemistry is hard, the marker genes for certain processes are a great shortcut, but unless you have the full biochemical pathway that you're hoping to find, marker genes are kind of useless, right? So there's a lot of pathways here that we can bring up and we can talk about in specifics, but I'm just going to stay general for right now, where you can have a marker gene that you look for. And you can find that marker gene and be like, yes, it definitely does this thing. And then as soon as you try and map out the rest of the process, because most marker genes don't stand alone on an island unto themselves, you realize that there are elements of either the cycle or the pathway that you're looking at are missing. And so you might have to then reconsider, is this pathway actually present in your genome, or is it some artifact that you can look into a little further? So lots of processes where the pathway is important and the marker gene doesn't tell the source story. That's what I'm talking about. And then when you shape your questions more or less accurately, you can do different searches here. You can do different questions. You can get a little bit, and some of them are easier than others. right? So the example I have here is, does this particular organism that I'm looking at have all the necessary and essential genes for nitrogen fixation? Is a much easier question to ask of your data than what role does this organism play in the nitrogen cycle, right? One of those is something that you can ask directly. You can build models, you can build databases, you can say, does it do nitrogen fixation? If you want to ask the question of, well, how does it fit into the nitrogen cycle? What transporters does it have? What reductases does it have? It becomes a much harder question. And that is a hard question to answer. And annotation is one, that step that you need to take and you need to figure it out. But it also requires you going back and rehashing, if you're not 100% familiar with it, the biochemistry that goes along with that question. So going back to KEGG, one of the tools that KEGG offers, so KEGG used to be a public database that you could go and you could download and you can do these searches on your own computer. Not too long ago, they've put it behind a paywall. It's very expensive. It's not very, that's not very exciting or user-friendly, but corporations have to, not corporations, but institutions have to make money. And if it wasn't being funded, that's how they do it. One way to get around that is they actually offer a service that you can use called Koala. So this is their in-house search tool that they use. There's a web page, which is not linked here, but I think is in the references, called Ghost Koala or Blast Koala, that allows you to take your protein information and search it against the entire KEGG database. And what they do is they give you back essentially a tab file of, here's the protein you gave us, that's the ID, and here is a function that we believe that this protein has. And so in our lab, we have a script that we use that parses out this information. So we've sat down and figured out what pathways you want to look at and what constitutes that pathway. And we can figure out a way of determining how complete something is. And it looks messy like this, but not the worst thing in the world. And it generates a heat map output like this that allows somebody to come in and look and assess if a particular function at a very general level is present or not present in the genome. And so this is available on on my personal GitHub page. And if anybody has any questions and wants to use it, they can let me know. It's also highly mutable. So if there are pathways that aren't listed here and it's something you're interested in, we can add those fairly quickly. And then you can get the search from output. And so, this example has carbon fixation and cytochromes, different types of oxygen cytochromes. It has sulfur pathways and nitrogen pathways and transporters and chemotaxis and things like that, things that we think about as being important in the marine environment. Another place to do something like that that has a lot of information and is a great service is IMG. So this is one of those services provided by the Joint Genome Institute where you can submit your data to IMG using this portal. It's not as easy as that. I say, oh, you can just submit your data. The Ghost Koala and Blast Koala, you can actually just submit proteins to and get an output. IMG, there's a process. You need to sign up for an account. You got to have clear approval. You got to submit your data in the pipeline that they want you to submit. It can take some time to run. They do them in batches. So they wait till they have a certain amount of data and then they process it all at once. So it could be a couple of weeks before you get your data back. But once you have it back, you have it in your own kind of little private area. And you can use the tools that are integrated into IMG to ask very broad questions about function of your organisms that you're working with. This is a little bit of a janky photo of the submission process through IMG. So if you want to submit, this is the link for that. They don't want you to giving them their raw data. And so it will go through and take your metagenomes and take your FASTA files and do these searches against all of these databases that we've been talking about and give you some predicted functionality that you can work with in that capacity. And then lastly, and this is one of those things, just to keep in your back of mind, this is the last slide for this presentation, for this part of the presentation, this idea that the more information you can provide when you submit data to these types of databases, the more useful it is for people going downstream. And you might hear that and say, yeah, but I don't want that data to be useful downstream to somebody else. It's my data. I'm going to keep it private. That's a horrible idea. guys, this is public money that you're most of the times using, right? If you're getting private money, that's fine. Do your own private money stuff. It's public money, public data. Get it out there. And so one of the things that you need to think about when you're taking data samples and collecting data off of it is what has been termed now is the minimum information for sequences, or mix. And so when you submit to JGI or IMG, they ask you some real basic information about where you collected your sample, its location, in this case, latitude, longitude. If it's a water sample, its depth. If it's on a mountain, its altitude. And then some. And at that point, it kind of slows down. But there's a lot of other information and categories that they allow you to fill in, including parameters that you've measured along the way while you're taking your sample: oxygen concentration, salinity. You can define the habitat that your samples are coming from. And what this allows your data to slowly make its way into the panoply of data that's out there, so that we can start making very large sweeping understandings of data and samples that are out there, right? Because as much and as useful it is to have your field of interest well characterized, if your field of interest is something that's been undergone agriculture, it would be maybe really useful to compare it to other fields that have undergone agricultural processing. So that way we can get at the heart of how does agriculture change the microbiota of a field that we're working in, right? And so these big questions require lots of data. And if your data doesn't have correct information part of it, it makes it really hard to find and be used later in the future. And, you're, like I said, maybe you don't want to share, maybe you're against that, but not all of us, we know the statistics. Not all of us are going to be PIs. Not all of us are going to continue on in academia for the rest of our lives, but we're the ones who are generating the data, right? A lot of the times. And, that data, if we don't do something with it, just gets lost. And so having this information, being able to archive it more accurately is a way to make sure that that data lives on, even if you don't continue to work in academia. But that's it. That was like my only soapbox moment of this entire presentation, but just to think about as you go forward. That will be it for our annotation segment. I hope it was informative. I hope JP doesn't mind me calling him out a couple times. And with that,